Folks, I'd like to say a few words about the importance of inhibition in the nervous system, and we'll encounter several applications of inhibition. You may recall inhibition reduces the potential for a nerve cell to fire a message. Now, notice that we'll be referencing inhibition quite a bit in these comments that I'll make. I want to talk a little about Bayesian inference. Bayesian inference refers to idea, it's kind of a top-down perceptual system, perceptual procedure in which high-level cortical coding may inhibit lower-level cortical coding in the interest of energy efficiency. Now, what that means is that when you have some kind of a perception, you see a railroad car, you see a watermelon, whatever it is you see. In other words, there's lots going on. Now you can see you color, you have form, you have movement, you have all kinds of these characteristics in the visual input that you have. Now, if a perception makes sense, in other words, if you see that thing, that really is a railroad locomotive, which is really complex. If you look at all the contours involved and all the shading, the different colors, etc. Well, if it makes sense, it might be a good idea to remove possible competing interpretations of sensations. In other words, we use our knowledge of the world in a top-down fashion in order to interpret new stimulation that we receive every day. In the brain, a lot of this is taken from our textbooks, from our reading material. Hierarchical processing compares interpretations of stimuli based on past experience. Now, I wrote that. But the idea is that, when we encounter some kind of, we'll use the visual system as an example. When we encounter some kind of visual situation, then we can say, well, gee whiz, that's recognizable. We don't literally say that to ourselves, but the idea is that our perceptual system is kind of pre-wired to use the information that's already stored. And therefore, we can kind of use the prior knowledge that we have in order to interpret new stimulation. Now, that's an example of Bayesian inference, in which, and there, the idea is that it's a top-down type of processing, right? And this is a quote from your textbook. Participants in a study viewed a series of visual images that all contained the same, set of lines, but at different spatial arrangements. In other words, you can imagine that you'd see a bunch of lines that are kind of randomly located, but sometimes they made sense in terms of like maybe seeing a cube shape or something like that in different orientations. And here's the research cited, Fang et al. In 2008 used fMRI. Now remember that fMRI is a technique to study the actual brain activity. It's not just a picture of the brain, but it shows real-time activity in the brain. Comparing activity in low-level and high-level visual cortex in the presence of different images. So the idea is if it's an unstructured image, in other words, it's a bunch of lines that really doesn't make coherent sense to most people, you're going to get more low-level cortical activity. In other words, within the cortex, right, which is not, which is just, a few millimeters thick. There's going to be thousands of nerve cells there. And the idea is that they kind of give way to the higher level processing, so that as the perception system is attempting to make sense of these random, lines at different orientations, well, if there's a decent interpretation available for those lines, then you can say, well, you don't have to use a lot of energy in the brain to produce an image, but you can kind of use information that's already kind of stored in the, in your perception system. For example, this is an image of a Necker, called a Necker cube. It looks like an ice cube where you have, a six-sided figure, and it's called a reversible figure because sometimes one side may be, maybe coming out toward you, and then later another side will be coming out toward you. The idea is these lines here, well, we can make sense of those. It was the first thing that you've ever seen. If you haven't never seen a cube like this, you may have difficulty making sense of these lines. They're not totally random, but imagine if the lines were just placed there in, literally in a random fashion. You might have difficulty coming up with a reasonable interpretation of what the image is. Right? But the idea is this top-down processing. In other words, this concept of the Bayesian inference says that if we're already familiar with something, that we can kind of make assumptions about the thing that we're viewing, and therefore we're able to come up with an interpretation without necessarily having to piece together all of the original pieces of information that we have. And your text, again, citing from your textbook here, the unstructured image evoked greater activity in low-level cortex. In other words, you're trying to make an observation, trying to make a reasonable inference about the information that we receive. On the other hand, the structured image, in other words, something that makes a lot of sense to begin with, it's easy to see, tended to go directly to the high-level cortex, and that actually inhibited, as far as we know, the lower level processing based on the fMRI data. And the important point is at the bottom here, once an interpretation has emerged, cortical feedback serves to suppress, that is to inhibit activity in lower level cortex that is not consistent with it. Now, so this is a top-down interpretation of how the perceptual system works. So that's just the idea that we often use past experience to interpret what we encounter new, right? In other words, daily interpretations that we have. Now, basically, notice that this is a cross section of the visual system. So light comes in, through the cornea here, and then through the lens, which, as we know, can undergo adaptation. In other words, the lens can become thicker or thinner, or I say not adaptation, accommodation. Right? So the lens becomes thicker and thinner based on how close an object is. So it's closer than about 6 meters away, the lens begins to be thicker. That's called accommodation in a normal eye, that is. And then the light comes in, and it stimulates the receptors on the back of the retina, right? And notice what it's an interesting phenomenon here, that these are the rods and cones, and if the actual location where we're focused on that image falls on a part of the retina, as we know, called the fovea. And there's a larger area around the fovea called the macula that contains, the fovea contains only cones. The macula contains, some rods. Remember that rods are for nighttime vision and cones are for daytime vision. Now notice that as the cones and rods are stimulated here on the back of the eye, on the back of the retina, this is the pigment epithelium here. Right? Now this is the very back of the retina, but notice that the rods and cones are in the back of the retina too, but they send information, toward the front of the retina. We're still in the back of the eye, but notice we have horizontal cells here. We have amacrine cells. We have several layers of cells, that is to say nerve cells, before the nerve cells collect and exit the eye in, what is this thing called? This is your blind spot where there are no receptors there. And this, of course, is called what? Right, it's called the optic nerve. And the optic nerve is made of the ganglion cells. Now, there's a lot of processing that happens, in the eye itself, based on the information coming from the rods and cones. And that has to do with, a lot of that has to do with this center surround antagonism, as we've encountered the concept before. Center surround antagonism is a common feature of sensory systems. This is, I believe, from your textbook. And it's involved with edge detection, as we'll see, in vision. But this, of course, is not vision. This example is from the touch system. In other words, in the skin. Now, what does this mean? You want to make sure that you're aware of what goes on here. In other words, the recording is done. There's some kind of a recording that's done higher up in the nervous system. In other words, not in the skin itself, but maybe in the spinal cord. Right? In other words, taking the information up into the brain. And notice that this is a center surround antagonistic receptive field here on the skin. See, so the idea is that now this is a tiny receptive field. This is enormously, magnified here in this particular picture. But notice that what this tells us is what a stimulation, where there's a minus sign here, stimulation will cause a reduction in the activity of the nerve cell. Maybe it's in the, in the spinal cord. if just the surrounding area is stimulated, but the center is not stimulated. If the center is stimulated, then you're going to get activity in the recorded nerve cell here, because there's a plus sign kind of in the middle of the receptive field. Now notice that the greatest, here's a little trace here on the right side, bottom right. The stimulus is at A, which would be, let's see, A would be in the surrounding field. Let's see, so the cell response, stimulus at A. Okay, the A would be the center. So when the stimulation is provided at the center of the receptive field, you're going to get lots of nerve activity here. This is the cell response here in the middle. If the stimulus is at B, that is to say, in the surrounding area, right? And then it's not very well labeled in the picture, but the idea is if the stimulus is presented at B, which is the surrounding where the negative sign is, well, that's going to shut off the nerve cell. But if the stimulus is at A and B together, you're going to get an intermediate response. You see why it's called center surround antagonism, because remember that the receptive field is considered to be on the receptive surface. Some people call it, part of the environmental area, right? But the idea is the receptive surface is going to be on the retina in the eye, or in the ear, right? Or in other words, the inner ear, or maybe in the on the skin, a patch of skin, right? But the nerve cell recording is actually up higher in the nervous system. And so the idea is so use the second half of the trace on the right side, compare B alone with A plus B. So notice when B alone is provided with stimulation, that would be the center, it shuts off the nerve cell. So we've seen this concept before, but that's the important idea. Now, in center surround antagonism, now notice that in the eye, for example, if this was not touch, but rather it was imagery, in the eye itself, this would be thousands and thousands of rods and cones on the receptive field in this particular receptive field, which is center surround antagonistic. Now, in the visual system, we could say, well, this is a ganglion cell. Let's say it's in the vision system. Now, notice this would be a on center off surround ganglion cell. Right, this is where this is the cell response. In other words, here is when the stimulus is presented. Now notice here's the on center off surround ganglion cell. So notice that here, the stimulation is provided only to the center, the target of the bullseye, right? The center of that receptive field. And look what happens. There's a lot of activity in the nerve cell that's being recorded in the optic nerve, maybe in the thalamus, right? Not in the retina itself necessarily. But notice that as there is no stimulation at all, right? It's darkness, right? Well, there's every nerve cell has kind of an ongoing low-level pattern of constant stimulation, and it's speeded up when it's stimulated. Now, notice as the size of the receptive field is covered entirely with light, well, notice that there's an intermediate level because you have cell responding in the nervous system. Why? Because this is an on-center off-surround ganglion cell. So here you're stimulating not just the center, but you're also stimulating the surrounding area. And therefore, you have inhibition and excitation going on here, which leads to, an intermediate level. There's a response, but it's not very big. Now, notice that because it's an on-center ganglion cell and it's an off-surround, by stimulating the surrounding area only, well, you're going to shut off the nerve cell. In other words, it's completely inhibited here. Now, this is the opposite. This is off-center, on-surround, right? So in this case, if the center is stimulated, center of the receptive field, look what happens. The nerve cell shuts off entirely. If the entire receptive field is covered, well, there's basically no effect at all. Just like you can see, if the entire cell is, when the entire cell is left alone, right? It's just like this entire receptive field is left alone. If the entire receptive field is simply not stimulated, well, you get that baseline of natural responding. And then here you got the inhibition being sent from the let's see, this is the off center. So inhibition is being sent from the periphery, and that kind of combines with the excitation for the, actually, it's, yeah, stimulating the center and the surround. Well, this is, if the center is surrounded, is stimulated, then you get the, the intermediate level of activity by itself, but then the surrounding area would be, the center is stimulated, the nerve cell shuts off, but the surround is stimulated, you get a lot of activity, which is what you have in the bottom trace here. You got the, the middle of the receptive field is not stimulated, but the surrounding area is. And therefore, because this is an off center on surround cell, it will show a lot of activity. It's a basic feature in these systems. And, for example, in the visual system. So here, kind of a schematic diagram of where these things are where these phenomena are occurring in our perception system. In the retina, as we know, this is a population code, right? In other words, there are three kinds of cones. You got the red cone, or the long wavelength cone. You have the medium level cone, which is the, the M cone, or the green yellow cone. You get the short cone, short wavelength cone. And that's what provides the trichromatic vision. Right? We say the trichromatic theory of vision. And then over here in the LGN, remember, this is the lateral geniculate nucleus of the thalamus. Right? In other words, this is the recording from the optic nerve, the ganglion cells that exit the eye and go into the thalamus in the brain. This is where you'd have, again, you get this center surround antagonism that are also called opponent cells, right? So in other words, you're recording from, the ganglion cell, and this is where you'll have your, remember we saw that green and red are antagonistic processes, just like blue and yellow are, as in the case of blue is the after image for a yellow stimulation, and green is the after image that occurs after constant stimulation with red. Now here you have the same opponent cells, the same opponent process idea, but they're incorporated into the receptive field, and we have these. In other words, scientists have found these types of cells. So in other words, green would stimulate the, what is that? Green inhibits this. This would be center surround antagonism. So you can have green inhibiting the activity of the cell that you're recording in the optic nerve, and then red excites it, right? So this would be, again, we have the color opponent system going on. Now in the higher up in the cortex, right, we can have like what are called double color opponent cells. So we'll get into a little bit later, in which the surround is a blue, medium wave. In other words, medium wavelength, or red inhibits and green excites, and then in the center, red stimulates and red stimulates and green inhibits. So you got these double color opponent cells going on, and they're called blob cells. Incidentally, we'll see a picture of those. The blob cells are kind of an odd word. They're in the cortex, and that's where you get your very fine shades of color process, not just red, green, and blue. Now, again, these are some examples of center surround antagonism. Right? So, in other words, you can have a green surround and a red center. Right? In other words, green excites and red inhibits, or maybe the reverse could be. But the point is, they're going to get a red-green system going on. Whatever it does in the surround, the opposite happens in the center. The same thing with blue and yellow, and black and white. Now, in addition, as we said, we have double opponent cells. Right? So in this case, it's not just that you might have a receptive field where green inhibits the nerve cell and red excites the nerve cell, but notice you can have double things going on. In other words, red inhibits and green excites in this case, right? And then over here, red excites and green inhibits. It gets kind of complicated, but the point is, you see why they're called double color opponent cells, double opponent cells. Lots going on. Now notice that because these are relatively difficult concepts to understand how to relate them to perception. But the idea is it's up to scientists to examine the phenomena that we know happens in the brain and to figure out how these characteristics of the nervous system are relevant to perception. So in other words, we know that these types of cells and this relationship between the receptive fields and the nerve cells higher up in the nervous system, we know that that relationship exists. Remember that a receptive field regulates or controls the firing of a nerve cell higher up in the nervous system. It's up to scientists to figure out, gee, how does that impact our perception? So it's up to us to kind of figure it out. Now this is a good example. This is we can say double color opponents in here. And I drew a couple. These are the same picture here. But notice that in this situation, you have a situation in which medium wavelengths, that would be green, would inhibit the nerve cell in the brain, whereas the long wavelength, and that would be a red, correspond to the red color, would excite the nerve cell in the nervous system. Now, based on this scheme, okay, now if you look at these, at the bottom half of the picture is double opponent cells. These are single opponent cells. So these receptive fields, notice these are in the cortex here. They're not in the eye itself, but the point is they're in the brain. Whether they're in they may be in the optic nerve, for example. Whereas the bottom picture is more likely to be higher up in the cortex. But look at this. You see the principle of opponency and how it can actually enhance contrast. In other words, if you look at the creature that's experienced this, imagine a chimpanzee looking for food, looking for fruit in a tree or something, right? Or a monkey or an ape, and notice they're able to detect the contour, the edge of the red fruit against the green leaf, for example. But notice that C, right, this receptive field would cause a response in a nerve cell in the brain that would appear brighter than B, right? Now this is B, this is C. So let's look at C. What's happening here? This is a red center excites the nerve cell, right? So notice that red is falling. This is the part of the fruit here, right? This is a red apple or whatever it is. You got lots of red. Now you got a little bit of inhibition from the surrounding area, right? Because it's also, well, this surrounding area is stimulated a little bit because it's actually going to inhibit the firing of the red. But notice that it's only partially stimulated. The outside, the surrounding area, the thousands of nerve cells, or I should say receptors that are stimulated here, are going to deliver inhibition, but not much, right? If you look at this subtle difference between nerve cell, that's nerve cell C, it's going to fire pretty, fairly a lot. It's not going to be inhibited so much. And notice that D is going to be very, very much stimulated. You see, this is the best stimulus. You see, so in other words, the one that's going to have a very strong, reaction. And notice that you're going to find, like over here. Now let's take a look at what's happening here. At B, you're going to have a lot of the green surround area here, right? So you're going to get a lot of inhibition there. You're going to get relatively little responding, because the center of the cell, which responds to the long wavelength of red, is not really stimulated at all, right? So you're going to get a real kind of a dampening effect, right? So in other words, it's going to cause inhibition from the green and excitation from the red, but there is no red. So you're gonna get a lot of inhibition here. And over here, you're gonna get a lot of inhibition, right? In A, right? Because you're gonna have what? You're gonna get you're going to get inhibition from the medium green wavelengths here, and there's no red at all, no long wavelength stimulus at all. So you're just going to get a very kind of a quiet nerve cell. It's not going to show much of a reaction at all. They're going to cancel each other. Over here, they're not going to be canceling each other because there's not so much inhibition from them. So it's going to look a little brighter. Maybe B would look, let's see, B would look maybe a little bit less inhibited than A, right? So you can see that this is the contrast. This is the edge of the leaf against, the contrast between leaf and red color fruit. So here's the worst response of the cell. Here's a better response, and even better, and even best, right? So in other words, as you move from the green to the red, you're getting more of a responding in the cell. So would C appear brighter than B? Probably, because you're going to get lots of stimulation from L. See, so in other words, you're going to get a contrast effect. And again, on we move to the bottom, we see, okay, does G appear brighter than F? And down here, let's look at what happens for, if the nerve cell that's controlled by this receptive field, what happens? Well, you have in the nervous system, this is the receptive field that's delivered to the retina of the eye. The surround is, stimulated by green. In other words, that's the best stimulus here. But notice they're going to kind of knock each other out. You see what I mean? In other words, the surround and center. That's why it's called center surround antagonism. They're going to kind of cancel each other out. So there's not much activity in the nerve cell. Now, what do you have here? This is inhibited by the long wavelength in the surround and inhibited by the medium wavelength in the center. So all you have here is inhibition. So you're going to get a great deal of inhibition. In other words, you're going to get more inhibition here than you are here, because in this case there's no because why? Because there's really nothing in the center which would be stimulated by red. Now here there's nothing stimulated by red in the center either, but because the surround is inhibited by the red long wavelength, you're going to get inhibition. So you may find that there's going to be maybe a dark part around the edge between the red and the, to enhance the contrast. Now, what do you find in G? Well, in this nerve cell here, that's controlled by this, this receptive field. Well, notice it's red. It's a long wavelength that's falling on the receptive field, and this receptive field is turned on. In other words, it turns on the nerve cell by the red color, and it's inhibited by the medium color, the green. So you're going to get a little bit of inhibition. From, why would you get a little inhibition? Because the surround is kind of stimulated a little bit by the long wavelength of red. But notice that you're going to get lots of inhibition from the centers, and then the surround is going to be at least partially green, right? So therefore it's going to be stimulated a little bit. So you're going to get a lot of stimulation here, because you got the long wavelength of red giving a little inhibition, but you still have a contribution of excitation from the medium. So the optimal stimulus for that receptive field would be, a circle, right? In other words, the surround is green and the center is red. And then over here at H, it may be similar. H and E may be similar. right? Because they're going to kind of cancel each other out, because you have, you have red stimulating the center, but the idea is you're going to get, you're going to get an inhibition from the surrounding red of the receptive field. Oops, I moved that around. I didn't mean to. Okay, there we go. See, so in other words, here you're going to have the surround is causing inhibition. The center is causing excitation. So you may actually get your brightest color. Let's see if we're correct here. You may get your brightest color when you get the strong firing pattern, because you get excitation in the center, and you get excitation from the surround also. So it may actually increase the contrast between red and green. So, this is, again, it's up to scientists to kind of determine, gee, what do these things have, how do they relate to our perception? But this concept of center surround antagonism seems to be central, and it's based on this idea of inhibition, right? So in other words, you have cells receiving inhibition from other nerve cells. So it may be involved with our, particularly with the perception of contrast, such as between red and green, and the same type of phenomena would apply to the contrast between, like, blue against yellow, because those are color opponent processes. Remember the color opponent processes: black and white, red and green, and blue and yellow, which is why as we mentioned, the after image for blue is yellow, and also the after image for yellow is blue in the negative after image. So to summarize some of that activity in sensory neurons is sometimes ambiguous, because sometimes you'll get several features going on in the same area, like the same receptive field. And notice that the nerve cells only have, they're called, univariant in terms of the messages they're able to deliver. They can only deliver the message of an action potential in a neurotransmitter. Now, to go just a little bit more into the population code idea, population codes can be made more efficient by using sparse coding, using targeted inhibition. In other words, kind of dampening out a lot of competing potential interpretations of stimuli. And some of the perceptual illusions are kind of interesting here. An illusion is something that doesn't accurately reflect reality, right? And under the topic of population, the importance of inhibition in population codes. Remember, a population code means that there's numerous kinds of cells that contribute to a perception, for example, of a color or brightness. And lateral inhibition means that you're inhibiting the nerve cell you're next to. One nerve cell inhibits the next nerve cell. One of those famous examples is called Mach bands, that we'll see in a few minutes. Now, notice that the alternative to population coding is called sparse coding, in which a very small number of neurons are able to encode a particular kind of stimulus configuration. Your textbook mentions, are there Jennifer Aniston cells, right? In other words, you can take a famous person, or maybe I think your textbook earlier mentioned your grandmother cell or something like that. In other words, your best friend, or your parents, or your grandmother, your grandfather. Is there a special nerve cell that you've trained to become active only when they see that person, right? Or you see a movie star, is there a nerve cell that registers Jennifer Aniston, right? And there's some evidence that it does happen. It's not totally compelling evidence, though, because it's a tremendous number of nerve cells we have, and so we're not really sure. But the idea is that are there possibilities that there's a single nerve cell that encodes your bicycle, or your friend, or something like that? Maybe not. we can't really say for sure. But that would be sparse coding. You see what I mean? In other words, you're coding for that particular individual using a very limited, number of nerve cells, maybe just one, very few. People haven't really gone into that in a lot of depth because of the difficulty in tracking down what all the nerve cells do. Lateral inhibition can remove the possibility of similar stimulation patterns, but not identical. What lateral inhibition does, it kind of leads to the perception of the major characteristic that you're looking at, such as in these mock band demonstration, like the contrast between neighboring or adjacent areas, as we saw in the fruit against the leaf analysis that we examined. Sparse coding, on the other hand, makes sense because there's a lot of energy expenditure in the brain, and it would make sense, to kind of devote a single nerve cell to just your grandmother, your Jennifer Aniston, something like that, because it would conserve less energy. It would really be a top-down approach because you're kind of activating a specialized, maybe single nerve cell. I kind of doubt it, but who knows? This is there are a couple of phenomena here. This is the mock band effect here. Now notice that explainable theoretically, as far as we know, by lateral inhibition. Now, if the effect works well for you, you may see a little bit of lightness in the lighter of the two bands, and you may see kind of an enhanced darkness in the darker strip. So, for example, let's take these two strips. In other words, the third and the third and the fourth from the right. If you look at these two bands, well, notice that the darker band maybe looks a little bit darker when it's next to the lighter band, right? And the lighter band may look a little bit lighter if it's next to the darker band. You see, so that's also linked to this simultaneous contrast effect, right? So in the simultaneous contrast effect, we can look at these two squares that are in the middle of those two, twin squares, right? And you may say to yourself, gee, this one on the right looks a little bit darker than this one on the left here, right? Well, that would be very reasonable. It is an illusion, right? Because in fact, they're the same. Just as, I think we've mentioned before, like this, these squares here, if you look at A and B, well, they're the same. They're literally the same. If you can block out, if you can block out the surrounding areas and just like through looking through a hole you made by bending your finger around, if you can somehow isolate just square A and B, you'll notice that if you remove everything else, they are the same brightnesses. They are the same colors in terms of black and white. This is the closer. I should have shown this earlier. This is the same thing. This is the mock band effect, right? So, for example, if you look at if the mock band effect works for you, let's take there's bands from the left, 1, 2, 3, 4, 5, and 6. If you look at just bands 3 and 4, well, if mock bands are apparent to you, then you will see, if you look at band 3, it looks a little bit darker along the edge where band 3 touches band 4. And band 4 would have its mock band where it looks a little bit lighter because it's next to the darker band. You see, now it's the same phenomenon we have here in these simultaneous contrast effects, as we see. Why is it called simultaneous contrast? Because you have two contrasts going on. Here on the left, you have black and a gray in the center, and over here you have another contrast. You have white in the surround, and then gray in the center. Now, what's happening theoretically here, right, is that you have, because you're going to stimulate a bright white along the outside area for the figure, for the black for the square on the right. got a lot of whiteness, brightness, a lot of light coming into your eye. And notice what that's going to do. It's going to deliver lots of lateral inhibition into the dark square, into the gray, right? And therefore the gray receives inhibition from the surrounding white area. Whereas on the left side, well, it's kind of the reverse. In other words, what you have is you have dark, black, dark color, right? Which indicates the presence of no light at all coming in to your retina in that part of your receptive field. And therefore, because these neurons are not stimulated around the edge where the dark black is, where the black is, right? Well, the square may look a little lighter because it's not receiving that inhibition from the surrounding black area. Here on the right again, well, the dark square receives a lot of inhibition from the lit white area in the surrounding area. This is called a Hermann grid, right? And if the illusion works for you, you see little kind of dark blotches at the corners of the squares. You may see a blotch here. In other words, the darkness, little dark spots at the edges, at the corners of the X's of the squares. Why would that happen? Well, here's the idea here is that lateral, remember lateral just means from left to right, right? In other words, not inhibiting the next nerve cell up, but inhibiting the nerve cells that are next door, right? It's lateral inhibition. So let's take a look here. You see, if the Hermann grid, let me go back to it. This is a larger example of the Hermann grid. And then this is the smaller version. You see, this is the little dots, the little black blotches you may see at the corners of the dark, of the dark squares. Now this is one of those corners of the dark squares. Now what's happening here? Well, over here, let's say here's ganglion cell number 1 here, right? If you can locate that here in the picture. Now notice that this is a center surround antagonistic receptive field. So you have lots of stimulation coming in from the center. And you do have you have inhibition coming from the surrounding area, right? And yet the inhibition it's not that much. It's coming from the right and the left. I'm referring to ganglion cell number 1 that's delivering, control over a nerve cell higher up in the nervous system. So the surrounding area delivers a little inhibition to the center, but not that much. Why? Because the top and bottom of the surrounding area are not being stimulated by light at all, right? And notice that the ganglion cell number 2 at the bottom of the picture, well, there's a lot of inhibition being delivered because it's like light will excite the center of the receptive field, but it'll inhibit the surround. But there's not much going on at all. There is no light in the center here, and therefore there simply isn't much activity at all. So that looks very dark, right? Now over here, now this is one of the the junction between the two white bands. In other words, these are the 4 the angles of the 4 squares. Where is there more lateral inhibition? The right field, right? In other words, the right visual field receives lateral inhibition from 4 sides. In other words, this visual field here, which is controlling ganglion cell number 3, right? Notice that this visual field is receiving, because the stimulation in the surround inhibits the center. And notice it's similar to this one, but in this one here, in the one on the left, the inhibition comes from the right and the left side, but here it's coming from the top and the bottom also. So the center is being inhibited quite a bit more in the right visual field in ganglion cell 3. There's lots more inhibition being delivered there, as opposed to ganglion cell number 1. You see, so when we look at the top, go back to the original Hermann grid, we can say, well, here we are, because we see the dark, kind of a darker blotch at the junction of the two white lines, right, that are adjacent to the square, you know, the edges of the squares, the right angles of the squares. So that's it. You see the darkness in the points between the squares, but not, you don't see the darkness in the larger white alleyways, if you want to call them that. You know, so that's a few, those are a few ideas about how lateral inhibition works. And it's the same here, because if you look at this one, you have lots of, you know, when you look at B here, let's analyze that. Let's see if this makes sense to us here. If you look at the square that has the letter B in it, and we look at the square that has the letter A in it, well, notice that the letter A is the square with letter A in it is receiving a lot of inhibition from from the top and from the sides. All right, that's square A. So in other words, inhibition is coming from the white squares adjacent to A, and also from the top where there is no square. So A looks a lot darker. If you look at square B, notice that square B is receiving inhibition. Is receiving inhibition in a very low amount, because the squares surrounding B are not only darker colored squares, but notice the shadow cast by this green cylinder, whatever it is, adds to the darkness. All right, so you got lots of brightness that's allowed to show up in B, but that brightness is kind of knocked out in square A because it's receiving lateral inhibition from the surrounding white or lighter areas. And I think we will stop this recording for now, and then we'll move into some color stuff and some movement stuff, and gamers' reflexes. If you're a gamer, are your reflexes a little bit better perhaps than if you're not a gamer? And the answer is yes. Okay.